You are all set.
Okay, so my title is Adapting to Climate Change Through Crop Improvement by Non-Sexual Gene Transfer. So some background, so climate change is just some recent ongoing rise in global average temperature near the Earth's surface, and it's caused mostly by increasing concentration of greenhouse gases in the atmosphere. So adaptation to climate change is basically the action of adjusting existing practices and processes in response to the threat of climate change. It's important to adjust because climate change greatly impacts agriculture, because agriculture is like very sensitive to the varying patterns of climate variations. So it's imperative to identify and evaluate the options for adapting to agriculture to climate change in the future. So some background on gene transfer. Gene transfer is the insertion of copies of a gene into a living cell in order to induce synthesis of a gene's product. So agriculture that's been gene transferred is selectively adapted crops who have desirable traits, who can achieve optimum yields while withstanding stresses like droughts and heat waves that are a result of global warming. So sexual methods of gene transfer are actually very unreliable because it's very laborious and time consuming. The natural process of recombination frequently will not separate the tightly linked genes, and therefore undesired traits that affect crop quality, yield, or adaptation may be carried along with the desired cell. And certain species of the plants that are characterized by strong heterozygosity and inbreeding depression don't benefit from sexual methods of gene transfer. So my proposal is to integrate beneficial traits into existing crops through the use of gene transfer by non-sexual methods. Um, so a method of non-sexual gene integration is also known as horizontal gene transfer, which is basically just transferring genes between organisms other than traditional reproduction. So the method of horizontal gene transfer involves first we need to be able to create a fully differentiated plant from non-sexual tissues and organs, basically regeneration. And in order to add a desired trait to the crop, a transgene of basically a foreign gene encoding the trait must be inserted into the plant cell along with basically a cassette of additional genetic material. The cassette includes DNA sequences called promoter, which determines where and when the foreign gene is expressed in the host, and a marker gene that allows breeders to determine which plants contain the inserted gene by screening or selection. A method of horizontal gene integration transfer is Agrobacterium tumefaciens. The use of a plant pathogen called Agrobacterium tumefaciens, which causes crown gall disease in many species, can be used. The bacterium has a plasmid or a loop of non-chromosomal DNA that contains tumor-inducing genes, also known as tDNA, along with additional genes that help the tDNA integrate into the host genome. For genetic engineering purposes, the agrobacterium must first be disarmed so that it does not make the plant sick because it is a tumor. Um, this is done by removing most of the DNA while leaving the left and right border sequences, which integrate a foreign gene into the genome of cultured plant cells. This has been done before, used with the nightshade family. However, um, it, it hasn't been used for more important crops like corn and beans. So the end goal of using horizontal gene transfer, particularly agrobacterium tumefaciens, is to profoundly influence the future of crop improvement through adaptation. The crops are expected to be genetically stable and useful, and the recipient plants varieties should remain unchanged except for the acquisition of new traits.
Questions?
So like the original methods where it causes like weird traits, like does it like directly affect
Yeah, it does. It's not guaranteed it'll affect the growth, but most likely.
So, pretty basic.
In essence, except for this is an really a newer method that hasn't been used, except for with nightshade.
What's sorry? What's nightshade?
Like tobacco tomatoes.
How would you think this would impact the current agricultural industry as opposed to current uses of GMOs? Do you think it will have a better impact?
This is like an alternate method, so there's not a lot known about it. So I'm thinking about like if you can test it, perhaps it's more reliable than the pre-existing methods. Yeah.
What's the difference between HGT and the sexual gene transfers?
What was that?
What's the difference between HGT and sexual gene transfer?
Sexual gene transfer is when you.
Can you go back to that slide and you address that?
This one?
Yeah.
Okay, horizontal gene transfer. It's so traditionally gene transfer is like hybrids, which you basically directly use sexual reproduction. But horizontal, you inject the gene into the cell.
And the method for doing this is what was the name of the bacteria?
Agrobacterial intimidation.
What's crown gall disease?
Oh, it's a
Say that
disease. Crown gall.
Crown gall disease.
It's a disease that the bacterium creates on the plant. It's basically a tumor, and from the tumor you get the genes that help integrate the tumor genes into the host genome.
When you say that this process is new, how new is this process?
I think it was around 1990, the process.
Okay. And again, so what you've read so far, you've seen it's only really been done in
Yeah, I saw it in Rutgers research.
tobacco and
The nightshade family.
and do you know what specifically how did they use this bacteria to modify the tobacco or the tomato crops? Like what traits or characteristics did they look at? What genes
They're
did they look
looking particularly for like resistances to herbicide and to weather extremes like drought and.
So we want tumor-inducing genes in the plants.
Yeah, you can disarm them. Yeah.
How would this process fare against diseases that basically just kill off GMOs? Is that essentially
This is DNA.
no against like diseases that would basically kill off the same type of GMO because they all have the same gene. They don't have defenses against
The GMOs. I don't know how they introduced the disease to the GMO, but any disease that would be on the modified plants with this method, they wouldn't have the disease because they would have been disarmed. So I don't think there'd be any disease to the GMO.
So stated that this method of gene transfer was only tested on one kind of plant. So do you have any certainty that you can apply it to like important crops?
That's the test. I'm hoping that it would result in like positive results, like they'd be resistant to heat and herbicide.
So the purpose of your research is to determine whether the gene transfer works with important crops.
Yeah.
Is this as cost effective as the regular sexual fertility?
Sexually?
Yeah.
Since this is relatively brand new, it's expensive. Using hybrids like sexually is less expensive.
Do you know why specifically they use a nightshade method, or we just tried it?
I don't know for sure, but I just know for a fact he tested on that shake and it was positive.